Ok, allora cominciamo questo meeting nel periodo post pandemia. Lo dico ridendo perché proprio oggi c'era la news della nuova variante, quindi siamo in qualche modo in questo momento molto delicato. Allora, la presentazione di oggi è fatta da un carissimo amico che ho conosciuto nel lontano, che ho incontrato nel 2004, dove abbiamo fatto per la prima volta un seminario, un workshop in Italia, a Napoli, su questa molecola che poi è anche stata la molecola cardine della sua carriera, che si chiama appunto NDPKB, o anche detta NME. E facemmo un seminario qui, due, ho dimenticato il numero, e facemmo un seminario qui per accogliere tutti gli scienziati del mondo che lavoravano su questa molecola. Allora eravamo 70. Ora, per quelli che stanno adesso sentendo, volevo discutervi del fatto che noi al tavolo discutevamo come si può diventare scienziati negli anni. Allora, ci sono due strade, quella di surfare sulle onde della conoscenza e quindi cercare le novità e continuare a raggiungerle e continuare a prendere la prossima onda. E questo è un modello di scienziato, cioè una persona che fa scienza guardando sempre la novità e cambiando da un posto all'altro l'onda. L'altro modello, che è il caso più o meno sia mio che anche suo, però lui adesso sta prendendo un'onda, è quello di andare sempre più deep, sempre più dentro al messaggio, per essere sempre più il top del top di quella materia, e quindi abbandonando quelle storie di onde, di argomenti che sono praticamente di voga. Allora, guardando la scienza in generale, è ovvio che dare una mission a uno scienziato è molto difficile, perché è un rischio per la vostra carriera di giovani qui al Cenge, oppure per tutti quelli che ci stanno ascoltando. Da studente dovrai diventare professore, posso mai decidere di studiare solamente quella particolare area? Allora, con Tom abbiamo trovato una soluzione, e oggi ve la racconterà, dove poi continui ad andare sull'onda partendo da un messaggio molto profondo. Il titolo del seminario di oggi di Tom è Inhibition of Nucleoside Dephosphate Kinases as a Novel Therapeutic Option in the Treatment of Earth Failure. E vedete l'ultima parola, Earth Failure, questo è un campo dove ci sono tanti finanziamenti e l'aspetto molecolare è in qualche modo chiave affinché questo campo possa crescere. Quindi in questa presentazione vi farà vedere Tom come, partendo da una molecola e facendo questo lavoro molto preciso, ha raggiunto poi dei risultati in questo settore. Please welcome to Tom. Penso che Tom ne parlerà in inglese. Sì. Quindi per coloro i quali stanno ascoltando e sono su Zoom, alzate la mano nel caso dovete fare le domande, io le riprenderò le domande per Tom, e quindi sarà un incontro che spiegherà la storia di questa molecola. Grazie Tom.
Sì, grazie Massimo anche per l'invitazione di presentare ancora qui in Napoli, ma penso che il mio italiano non è sufficiente per una presentazione scientifica. Per questo I switch language better to English, because I feel much more familiar in scientific terms in English. So I apologize for not speaking Italian to you, but I like that language, and it's really nice to speak Italian, and we can speak that together in a personal way, but not for my presentation. So as Massimo already pointed out, so my interest with that molecule goes back quite some years. So to be honest, it started during my PhD thesis. So, and the first couple of data slides are originating from that, and you will see that. Okay, so, but first, before we start, should move forward with the arrows. Oh, here.
Yes. That's right.
Okay. Now it's working. Thank you very much. So first, a little bit about my pet enzyme. Many of you know that, but I think as we have also outside these people listening to that, I just want to present you this enzyme, which actually got the wrong name, because nucleoside diphosphate kinase would imply that the kinase is doing a job, what you all know, phosphorylating other proteins. Indeed, it does it. We'll later come to that, but it was originally discovered in another mode. And what it does is, to be in a biochemical way speaking, NDP transphosphorylase. So it picks up a high energetic phosphate mostly from ATP, because ATP is the general energy donor in a cell, and then transfer that phosphate on a histidine residue within its enzymatic cleft, and then the ADP, which is formed from that, leaves, and another diphosphate can enter the cleft and pick up the high energetic phosphate to form another triphosphate. And this can be either a desoxynucleotide or a ribonucleotide, doesn't matter. So the enzyme is, of course, housekeeping in the required for the homeostasis of nucleotide in every living cell. So even the slowest bacteria have NDPKs. Yeah. The enzyme which I will talk about today is mammalian isoforms, and mammalian isoforms are usually hexamers built of 6 enzymatic active subunits. So there are 10 members of the NME family. NME is the genetic name. Meanwhile, it stands for non-metastatic enzyme, which comes from another field, but the name has been adapted for all of them. 4 of these NME genes belong to the class 1, and these are A, B, and C, and D, or 1, 2, 3, and 4. If you talk about the genes 1, 2, 3, and 4, about the enzymes NDPK A, B, C, and D. All of them have enzymatic activities. If you look at on the expression level, then the 2 major isoforms A and B are highly abundant, practically present in every cell. NDPKC is expressed at lower levels, and NDPKD has a specific function in mitochondria because it is a mitochondria-targeted protein, which has their functions, which I will not touch today. But as I mentioned before, that these are hexamers, and the interesting thing is that these are not, these hexamers can be oligomeric. It means that it's heterooligomers, that it's not made one of 6A subunits or 6B subunits or 6C subunits. This happens, but if you look closely, most of these are heterooligomers, which are made mainly of A and B, but C can also enter that oligomer, and that will become important later on. So, and the second enzymatic activity is indeed now a kinase activity, which these enzymes have. They are bona fide the first histidine kinases, which has been found in mammalians. And the scheme is quite the same. So you can, the ATP or the triphosphate, or could also be from GTP, is picked up by the enzyme from this intermediate histidine phosphorylated form. But then it can also use a protein as substrate and transfer the high energetic phosphate onto another histidine residue within that protein, and then you have histidine phosphorylated protein. So that's the two functions of that enzyme. So, and both will be important later on. So I promised you some slides back from my PhD, and that's how I got in that field. You know that scheme on the left here, you know most likely pretty well. That's an activation scheme of a heterodimeric G protein driven by a GPCR. So if a ligand binds to the GPCR, an agonist, then it will release GDP from the heterotrimer, so GTP can bind, and then the heterotrimer dissociates, can regulate effectors. Then there's a biochemical clock inbuilt, so the alpha subunit hydrolyzes the GTP, GDP is formed again, and then this heterotrimer reassociates and the cycle starts over. And this can do several times. And by every time it turns, one GTP is used by the cell. So there is a need for replenishment of GTP at the site of action. Otherwise, at these sites where the Ig protein content is, the cell will be depleted out of the nucleotide. And that's where the enzyme comes in play. So the general idea at the beginning was that NDPK can use ATP and the GDP produced by the G proteins to replenish the GTP and then provide energy for the activation of G proteins. And I have to mention that most of the isoforms of NDPKs are cytosolic, yeah? But already in the beginning, there was some evidence that there is a membranous form. And that is shown here. In this case, we prepared membranes from human platelets. So, and they contain, of course, the G proteins and the GPCRs. And then we took advantage of a very elegant way to prove the activity, because we had to stop somehow the use up of the GTP by the GTPase. Otherwise, you will not catch the intermediate. But there's another ATP analog, which is ATP gamma S. So the gamma phosphate carries one sulfur instead of an oxygen. This is usually stable nucleotide, so it's not degraded by ATPases and other ATP-using enzymes, but it can be picked up by the nucleoside phosphokinase and transfer the thiophosphate, in this case, then back on GDP, and then you get a formation of GTP gamma S. This is a GTP analog. But this is, again, very stable and is not hydrolyzed by the GTPase of the alpha subunit, so which will cause a quantitative activation of the G protein. And in these membranes, as you can see here, if you provide ATP gamma S, and then we used radiolated GDP, you get a formation of radiolated GDP gamma S. If you put an enzyme, and this is creatine phosphate creatine kinase, which competes for the GDP as a substrate, then you repress that, because then GTP gas cannot be formed. And that this GTP gamma S is indeed able to activate G protein is shown on the left side. Here we have either a condition with magnesium or magnesium with ATPase. Magnesium is an important cofactor for the NDPKs. They will not work without it. So if then you incubate the membranes just with ATP gamma S and magnesium, you already get an basal activation of the G protein. If you add an agonist, which will speed up the release of GDP from the G proteins, you will clearly enhance that, because the GTP gamma S can enter. And in this case, also here, the CPCK as a competing enzyme suppresses that. So that is the action you see here is also given in the activation of the G protein. So there is a way that these enzymes are related. They are in the membrane, and you cannot be washed out. So the nature of that association took us a while to get there. So, but we found out that NDPK, especially one isoform, the NDPKB, forms a stable complex with the beta-gamma subunit of the G protein. And it does even more. I told you that it is a histidine kinase, in addition to being a NTP transphosphorylase. So when we immunoprecipitated G beta gamma with the G beta 1 antibody, you see that here, and then incubated that with GTP as a phosphate donor, this is a radioactive GTP, so the gamma phosphate is a P32, and then make an autoradiograph. You see here the precipitated G beta, but not with the antibody. It's highlighted by a phosphorylation, by phosphate, and you also see the NDPK down here in that complex. It's not precipitated if you use a control antibody. So we identified the histidine residue, which was phosphorylated in G beta by NDPK, and this is histidine 266 in G beta. And we wanted to know whether this plays a role in the activation of G protein. So I just give a cascade here. So if you use ATP, then you get NDPK intermediate phosphorylated, then we get a phosphate on histidine. And if this has something to do with, is required for the actin of GGP, G protein, in this case GS, then it should then lead to the activation of formation of cyclic AMP and cellular effects. So what we did is we mutated this histidine 266 into a leucine, made an adenovirus, which then within a cell would express beta 1 gamma 2 as a specific subunit, and you can increase the expression of this beta gamma dimer by increasing the viral load on the cells, and then you can clearly see here that we get the expression of that protein, wild type or mutant, in a similar way. So then we looked what is downstream of here. So we measured cyclic AMP. In this case, in the cells we did that was adult red cardiac myocytes. And as you can see here, this is a control virus. Where's my pointer? Here. This is a control virus. This is cyclic AMP formation, basal cyclic AMP formation. This is the overexpression of the wild type protein. There's no significant difference, but if we use the mutant, which will not allow this reaction, then we see a significant reduction of the basal cyclic AMP formation by about 50%. And if we then measure the contractility of these cells by single cell shortening of isolated myocytes, you see the difference. So this is a control virus. This is the wild type G beta gamma. If anything, it increases a little bit the basal contractility, whereas the mutant, which will not allow for the transfer of the phosphate, then you see a decrease in that. So somehow this pathway is involved in a receptor-independent basal activation of G proteins by nucleoside kinases. So, but the story turned out to be a little bit more complex. We detected NDPKB in that complex. This is one of the two major isoforms. At the time we did that, nobody was talking about NDPKC a lot because it was not on the radar. But we struggled a long time because what we tried is we purified transducing G beta gamma. Transducing is the G protein out of the retina. And this is the only G protein you can get in soluble form. All the others are membrane bound because they are modified on the gamma subunit in a way that you need detergent to extract it from membranes. Transducing you can extract that way. And when we combine that with NDPKB in purified form, or here in overexpression in cellular form, and there was no interaction between these two. This is nearly blank. There is no interaction. If there would be an interaction, then NDPKB would bind here, and then you could detect that with an antibody against NDPKB, there's no bind. Instead, and this was the surprise, if we used NDPKC, a complete different form. So we overexpressed NDPKC in the flag-tag form in red cardiac myocytes, took the lysates, spotted transducin on the membrane, and then indeed it was fished out by transducing the NDPKC. So this isoform binds. And if we do an experiment where we spotted NDPKC on the membrane, purified NDPKB on the membrane, then this is also able to retain NDPKB with signing of a heterooligomerization of these isoforms. If this is true, then we should use NDPKC to get also binding of NDPKB to the G protein. That's an experiment we did here. So this is the same condition here. We overexpressed NDPKB and in the cells, and then spiked in increasing amounts of NDPKC in that overexpression. And then we studied the binding of NDPKB and not NDPKC to the transducin. And as you can see, as more NDPKC we bring into the cell, as more NDPKB is now going to bind. So we have the heterooligomer, which is binding to that. And this is not only true for transducin, but you can do similar experiments. So G beta 1 as a complex partner should bind, but also G alpha S and G alpha I2, which are found in the heterotrimers, depending what you're looking at, can also bind. So you have complexation of NDPKBC with every of these two of these major G proteins. And this is a final slide here, experiment here, which you really use purified recombinant proteins to see that they interact in the test tube. So we purified transducin, we made recombinant NDPKC, in this case with a his tag. A his tag can be bound to a nickel NTA matrix, and if transducin will bind to NDPKC, it should be co-eluted from the matrix if we use imidazole to release the NDPKC from the matrix. And that's exactly what you see here. This is transducin passed over the column without NDPKC, and there's no elution of transducin, so it passes all through. Here is NDPKC shown here. And if we elute that NDPKC together, which has seen transducin, then it's co-eluting. So it's really binding the two proteins. There is nothing in between because this is very pure proteins. So I will stop here with the pure biochemistry because that's something which is done in vitro. It's just does not say anything about the function of that molecule. So we wanted to know whether that is really important for life or for the heart function, because everything what we studied so far was related to the heart, because my heart is going there research-wise. So I thought, well, if we would do that by my mouse research, we're dealing with 5 G beta subunits. We're dealing with 12 gamma subunits. We're dealing at least with 3 isoforms of NDPK. In this case, this will take forever to study such things in a mouse setting. And at that time, I had a collaboration with partner in Heidelberg, and I was aware that they study these such protein-protein interactions in zebrafish embryos. And zebrafish embryos can easily be manipulated by a technique which is similar to what you all know for siRNA, suppression of the expression of a certain protein, and that you don't do with siRNA in this case, but you do that with so-called antisense morpholinos, which will bind the RNA of the mRNA of the protein and will which will not be expressed and thereby lower the expression of a certain protein. And that we did for NDPKs and G beta. And I show you just how efficient this is. Here is an expression of NDPKs, in this case, detected by non-specific NDPK antibodies, so it recognizes all 4+1 isoforms. As you can see, this is the regular expression. If we take a morpholino against the A isoform, you see a reduction. And also if we take a morpholino against the B isoform, you also see a strong reduction. That the morpholinos are specific, working is shown here. Then we use here an antibody which is only recognizing NDPKB. The A knockdown does not do anything on the expression of B, but the B knockdown completely suppresses the expression, at least on the level of the immunoblot of B. So, and this can be also shown functionally. If you measure the activity of the NDPK and cell lysates, then you see that both the A knockdown or the B knockdown reduces the cellular activity around 70%. So, and the zebrafish has one very nice feature for us, the zebrafish embryo, because you can see its heart working as an embryo. It's translucent. You can fix the fish in a gelatin matrix. It can live there. And then you just do a video and look how the heart is doing. And I'm showing you that here. Should work. Now you see here, there's a 2-chamber heart in the fish. And here's the atrium, and here's the ventricle. And if you look closely, this is the regular beating. And you see even the erythrocytes pumped through that heart. So now I show you the same experiment with an NDPKB knockdown. The A knockdown doesn't show any phenotype. The B knockdown, you see how this heart is impaired. The atrium is beating a little bit, but the ventricle is completely silent. And we get exactly the same phenotype if we knock out the interaction partner G-beta-1, which is part of the G-beta gamma subunit. The fish has 2 isoforms of G-beta-1, both of them is doing it. Okay, so following from that experiment, we should get a similar result if we tackle NDPKC instead of NDPKA. This is shown in the next slide. Again here, this is a wild type control morpholino. There's no alteration in the fish. If you knock down NDPKC, which is shown in the Western blot here, then you get the similar impression. And if you look at the heart again, so again, similar, the atrium is still beating, but the ventricle is nearly silent. And this you can also measure. If you measure the fractional shortening here of that ventricle, it goes down with the knockdown, you see clearly, whereas the wild type is not attenuated. And this is related to cyclic AMP formation. If we measure cyclic AMP formation in fish lysates, you see that the knockdown is reducing that by around 70%. And that's the same if you do that with the B knockdown. And we even did that with isolated zebrafish heart. We collected them and measured cyclic AMP in this tiny heart. So we have to pool them, hundreds of them, but you see the same. So you have to believe me, that's how it works. Okay, but also zebrafish is far away from that what we are talking about. You want a title I pronounced was talking about heart failure. So that's a human disease. And that's a major disease in elderly. So is there something we can ground something on that NDPKs has to do something with that also in humans? Okay, we had access to tissues from human hearts, from failing human hearts, and from non-transplanted, non-failing human hearts. So they have been selected for certain reasons why they have not been transplanted, but it's the best what you can get as a control. Getting the failing heart samples is not the problem, because the surgeons take them out and for them it's waste. And but the transplanted heart, that's the rare issue. And if you need to get some hands on that tissue to have a control. Okay, we did that. And we wanted to analyze the complex formation in that heart. And so what we did is we did lysates of that heart, and we we performed immunoprecipitations. We immunoprecipitated with antibodies against G-alpha S, G-alpha I2, I showed you they bind, and also with NDPKC because it's the interaction partner. And as you can see here, this is on this side non-failing hearts, and these are the failing hearts. You see, if you precipitate G-alpha S, there's NDPKC bound to G-alpha S, and in the failing hearts, it's less. If you look on G-alpha I2, that's the inhibitory G protein, which will repress cyclic AMP, which is a well-known feature of what occurs in heart failure cells, then you see, okay, here there's nearly no binding of G-alpha I in the non-failing hearts, but an increase in the failing hearts. And the same is reflected here if you use NDPKC as a precipitation, and then you look for G-alpha I2, it's increased in the failing hearts, and in G-alpha S is reduced in the failing hearts, whereas in non-failing hearts, that's the standard. So, okay, that's an association. We wanted to know whether that has functional consequences. This we cannot test again in humans because we cannot use human cells for that. So we used in this case neonatal red cardiac myocytes, you saw them before, where we can adenovirally overexpress certain proteins. And first what we overexpressed is NDPKC, and then we changed the GS-GI ratio, which is occurring in heart failure. In heart failure, it's known that GI is going up and or GS is going down. There's a discussion, what is really the case if the one is going up or the other is going down, but the ratio is definitely different. So what you can see here, if we overexpress NDPKC alone and then measure the cyclic AMP formation in these cells with a beta adrenoceptor agonist, which will stimulate the beta adrenoceptors, will activate GS, and then turn cyclic AMP formation on. You see that the stimulation of the GPCR is doing that what you expect. You see a large increase in the cyclic AMP formation. And if we add NDPKC, then you see even more cyclic AMP formation because that's most likely the formation of a complex with GS. So now we alter the ratio. So we overexpress GI alone, that's here. Then GI is alone and it binds GTP, so it's getting constitutively active, and then it represses cyclic AMP formation. And the interesting thing happens then in the combination. Now, it's going further down if you add NDPKC in the scene. So by changing the ratio between GS and GI, NDPKC picks that one, which is more predominant, that's you see also here, and it's acting on that G protein and therefore increasing the suppression of cyclic AMP formation. And that's exactly what you see in heart failure patients. If you measure cyclic AMP in these hearts, it's always chronically suppressed. So the idea we got from that we had to tackle that. And I just want to summarize that for you before we go in the next part of the talk. So NDPK supply G proteins with GTP from ATP and GDP, that's obvious. The isoform NDPK B and C form a complex with G protein beta gamma subunits via an intermediate histidine phosphorylation of G beta. This complex modulates basal cyclic AMP formation and contractility in cardiac myocytes. And in human heart failure, the expression of NDPKC, which is upregulated, also like GI, then alters the prevalence of G proteins found in the complex with NDPKA. So see a shift of the NDPKC from GS to GI. And this prevalence is likely responsible for the reduced cyclic AMP formation seen in the heart failure patient, or at least in part. So the idea would be that in the patient, you would need some substances which inhibit the activity of NDPKC to counteract this suppression of cyclic AMP formation and regain contractility. And that's something we wanted to test, and we looked for inhibitors of NDPKB and or NDPKC. So we did a screen together with Sanofi in a drug compository they had. And this has been pre-selected for potential histidine kinase inhibitors because we thought that might be an idea to look at that. And from these more than 1,000 drug compounds we screened there in an assay, we found 3 which did suppression. And just let me explain how the screen works. In this case, we don't measure GTP formation, we measure ATP formation from GTP and ATP. And this has the reason is that the ATP formed can be used by a luciferase and can be transformed into light. So if ATP is formed, we get a light signal, and this we can do in a 396 well plate and screen a lot of compounds. And that's what we did. And we found that 3 substances, and one of them was very promising. This is what we call SUNV3. It's a substance which is soluble in water up to 100 micromolar, and you can see how it works. This is just the ATP formation without the inhibitor, then increasing concentrations. We pharmacologists do that over a log scale because otherwise the curves doesn't really show how the affinity is. But here you can see that because you get an IC50 out of that, how the infinity most likely in which range it is. So, and we used in this case purified NDPK, purified NDPKB, and purified NDPKC. These are coming out human proteins recombinantly formed in E. coli, but these are done homohexamers, so pures. And as you can see, there is slight inhibition of A, but at only very high concentration. There is a decent inhibition of B, but most prominently C is inhibited by this substance. Okay, that was very interesting for us. And then we wanted to ask the question, what kind of inhibitor is that? So from the enzymatic way, you have 2 ways. Either it's competing with the ATP or GTP as a substrate, or it's altering the enzymatic activity of the enzyme itself, and then it's called an allosteric inhibitor. And to this, you can dissect that if you measure the enzymatic activity at increasing concentrations of the triphosphate donor, in this case GTP. If it's a competitive inhibitor, as more triphosphate you add, as less the inhibition should be, and it should be completely gone if you have 10-fold more than the inhibition constant of your inhibitor. Oh, that was too fast. But if it's an allosteric inhibitor, it will inhibit at any concentration in a similar mode. And that's exactly what is happening here. If you increase the concentration of the triphosphate, you see more enzymatic activity, but the extent of inhibition is always the same. So it's bona fide an allosteric inhibitor. And what is very nice for us, it's also working in cells, it's uptaken. So in this case, we just overexpressed NDPKC in a hex cell model, and then visualized the intermediate phosphorylated form of the NDPKC by using an anti-phosphohistidine antibody from the lab of Tony Hunter. That's the P1, the N1 antibody, which is recognizing typically the form of NDPKs. And you see this is the autophosphorylation of the NDPKC. And if you add the inhibitor, it suppresses that. So before going into the more experiments with that substance, we wanted to know where does it bind? How does it function? And there's one method to tackle that without crystallography, because crystallography takes a lot of effort. So this is the hydrogen deuterium exchange mass spectrometry. What you do, you take your protein and then you incubate it with and without your ligand with deuterium. And by an exchange rate, every hydrogen on the surface of the protein will be exchanged against deuterium, which is another molecular weight. So, and if you then digest the protein into little peptides and put them into mass spectrometry, you see all the deuteriated peptides. There where the ligand is binding, the exchange is not possible. They are protected, these hydrogens, and then you see there the hydrogen, and in the mass spectrometry, then there you know on your molecule where the binding is. And that's what we did. You see here in green, that's the unligated NDPKC, and all in blue are protected regions. And together with Wieland Steinchen and Gerd Bange in Marburg, we modeled that onto the NDPKC monomer crystal structure, which is available in the protein data bank. And what's coming up is, you see here, this is the enzymatic cleft, where in this case ADP is bound. And the potential binding sites are all around here, this cleft nearby. So that's obviously where it could bind. And we teamed up with a modeler who can model our small compound into the amino acids into this cleft. And it's the only thing which I'm not allowed to show for IP reason is the structure of that inhibitor. So that's why you see this blurred banana here. But that's exactly the out shape of the molecule. Yeah, it's binding here in that cleft and an NDPKC, and there are interaction sites. We know which amino acids have polar, unpolar interactions. So it seems likely that this is indeed, and it's sitting next to the ATP binding site, the enzymatic cleft. So it's also the allosteric inhibition we observed makes sense. And then another feature that we really hit the structure is given here. And we have found in a compound library, which you can buy online, there are similar molecules with analogous structures. You can just buy them in small amounts. And we found out that these NDPK, there's one compound which has a nitration on a benzene ring in the molecule. So we bought that compound and tested it also. And so nitration is a very strong alteration in the structure. So what then happened is that we changed the preference of the inhibitor. In this case, again, NDPKA is only slightly inhibited, but also NDPKC is only slightly inhibited by that inhibitor. But this now has a preference for B. So this structure is somehow really on top of NDPK. So that makes us promising. Okay, and to make a long story short, we tested the inhibitor against other kinases which are known. We had a panel of 16 serine, threonine, tyrosine kinases. It does not inhibit, so it's specific. And we also looked in a pharmacological screen against 30 other targets like GPCRs, channels, and so on, and could not find a confirmable hit. So it seems to be rather specific for NDPKs. Okay, so now we have the inhibitor. Now we want to see what it's doing in heart cells. Yeah, and heart cells are not easy cells. It's not like a plastic cell where there's a receptor and then you measure cyclic AMP and then you see what is going on. They are compartmentalized. So the cyclic AMP action within these cells are rather specific for subdomains. And I show here this the scheme again. This is a beta adrenoceptor. It's mostly beta 1, but also beta 2 plays a role. This is GS adenylyl cyclase, cyclic AMP is formed, PKA is activated, and then there are quite a bunch of PKA substrates which are involved in the contractility. There's, for instance, it's not on the scheme, the L-type calcium channel, which lets the calcium into the cell. It's phosphorylated by PKA. Very important subdomain is the Serca domain, is the macroplasmatic reticulums with two subdomains, the ryanodine receptor, which on the enter of calcium releases more calcium, and this is regulated by the phosphorylation, and the reuptake of calcium by the circa, the calcium pump, is also regulated by phosphorylation of accessory proteins. And then also the contractility of the muscle itself is on the fibers also regulated by phosphorylation of, for example, troponin I and other proteins in the contractile apparatus. So how to access that now in a more distinct way, not only measuring cyclic AMP as a cumulative result, breaking up millions of cells, and then measures cyclic AMP. So we teamed up with the lab of Vyacheslav Nikolaev, because he is an expert on so-called FRET experiments, first resonance energy transfer. And this is an optical method which allows to measure the distances between molecules. It works in a way you have two chromophores. There's a CFP, which is excited by 436 nanometers. And if a YFP is in a plausible distance, very small distance to that CFP, then the energy can be transferred on the YFP, and you see an emission in the wavelength of YFP. So, and they link both CFP and YFP by a cyclic AMP binding domain out of a protein called EPAC. And with this sensor, without cyclic AMP, it's folded, and then the energy transfer can happen. If it's binding cyclic AMP, then the molecule is stretched, and the YFP moves out away from the CFP, and the FRET goes down. So this working is showing here. That's the cytosolic form of that sensor. So these are the cells. These are mouse cells, cardiomyocytes from a transgenic mouse, which express this permanently, this sensor. So these adult mouse myocytes, if you add isoproterenol, which will activate your beta receptor, you see what you want to do. What you should see, that by cyclic AMP formation, the sensor moves away, and then you see a lowering in the ratio. And then to level that out and get a relative relation to the total amount of cyclic AMP available in such a source, you added an inhibitor of phosphodiesterase, which will stop the degradation of cyclic AMP, and phospholin, which is a direct activator of the cyclase. So this will give you the 100% signal. And as you can see here, that gives a very strong reduction in cyclic AMP. So you can always relate your response to what you see here in isoproterenol. And that's a cytosolic form. So the black one is a cell without our inhibitor. The red one is a cell with our inhibitor. There is no difference in the cytosolic form. Okay. So, but what is the nice thing of that? You can target that sensor by binding to other proteins into several subdomains within the cell. So you see here one sample where it's an associated protein to the L-type calcium channel. This highlights the plasma membrane domain, as you can see here. Then there is a junctional protein which is related to the ryanodine receptor microdomain, and there's another protein which targets that into the circa domain where the uptake takes part. Okay, and now we can compare. By using these sensors, you see the different pattern, how it looks like. We can compare the effects of the inhibitor on the subdomains in the cell. And this is now, again, the plasma membrane. There is no influence. You see the increase by ISO is 40% increase. It's not altered by the inhibitor. The control here in the ryanodine receptor domain, and also this SANV3, no change. But then came the surprise. When we looked at the circa subdomain, we saw that the inhibitor is increasing the cyclic AMP signal in that specific subdomain by about 50%. And that this has really functional consequences is shown here because we have measured in these cells the phosphorylation of phospholamban, that's the pKa target, and in the circular domain. And as you can see here, this is what ISO does is alone, and this is the combination, that's a clear increase. Okay, that is interesting because Selker was always a target in heart failure therapy. There were people in Canada which produced an adenoviral construct to overexpress that in models of failing hearts. And they even got this with an AAV virus into a phase 1 trial in humans. But it failed because of regulatory reasons. So, but the domain is very interesting. So we thought, okay, if our inhibitor is doing something in that domain, we should see that in calcium handling because that's the important measure. So there are several aspects you can study there. And we used here together with Lars Meyer and Karin Hammer in Regensburg, again, mouse cardiac myocyte, and we measured different parts in the calcium cycle during electrostimulation of such myocytes. And that's something you can, I have to say, that's not with an agonist, that's basal, only electrostimulated. They release that on electrostimulation, the calcium, take it up and again, but no agonist there. So what is important is, for example, this SR calcium load. If you have more calcium in load than on an action potential, there can be more calcium released from the sarcoplasmatic reticulum, and this will increase the contraction. And this is exactly what we see if we measure the SR calcium load, it's increased, and if we also measure the calcium transient, which is in this case released by electrostimulation, we see a significant increase. The diastolic calcium is not altered. That's very important because the diastolic calcium is always important to keep the cell quiet. If you have an increase in diastolic calcium, there's a tendency to arrhythmias in these cells. What we don't see in that case here without an agonist is really an increase in contraction as measured by the sarcomere length of that cell. So if we should really see an increase in contraction, then we would see an increase, a shortening in the sarcomere length. There is a tendency, but it's not there. Yeah. At least in these experiments. So we said, okay, we have to go more to in vivo. And the next thing what we did is we used rat heart muscle strips, put them in an organ bath, also electrostimulated. And you see, if you see it here, you see, if you look closely, you see the muscle contracting from time to time. See it? It switches. And there's a spring down here, and this is electrically recorded, so you can measure force. So you can measure the force of that, how the muscle contracts. Okay, and then you can add in such an organ bath substances. You can either add isoproterenol as a beta agonist, and it does what it should. It increases contractility in a concentration-dependent manner, and you see the maximum was reached by 30 to 100 nanomolar. That's exactly what you see is known from the literature. And then if you add that, our inhibitor, now you should see more contraction if this is the case. And that's exactly what we do. So it seems that the inhibitor is sensitizing the muscle for contraction. Also, this is very nice, very promising. But again, this is ex vivo, yeah? So you will never see an organ bath in a patient. So we cannot go to patients, but we can go to mice. So, and the easiest thing first, you don't use a disease model. You first have to use a healthy mouse, whether it would tolerate the substance at all. And so what we did, we took some 2-month-old mice and injected them for 7 days with a daily IP injection of 100 microliter of 100 micromolar of our inhibitor in PBS, and control mice only with PBS. And the mice were perfectly healthy. No sign of any toxicity, no changes in some organ functions. They were like wild-type mice behaving. And that they ate, we took the mice out and made an echocardiography on that to measure the heart function. And after that, then we sacrificed the animals, make an organ collection to look for the changes in liver, vessels, and so on. And for example, we took also out the blood, made plasma out of that. And by mass spectrometry, we could measure the concentration of our agonist, of our inhibitor in that blood. With using the substance as a control, the pure substance, we could calculate that we have a range between 2 and 3 micromolar of that substance in the blood of the mice at that time point. No obvious phenotype. So then I was not expecting anything out of that because it's a healthy mouse. Yeah, so my first concern was, would they tolerate that? Would they die, or would I see any dropouts? Would I see some toxicity in the liver or something like that? Nothing happens. Then we measured the heart rate by that echocardiography, no change. The LV diameter of the left ventricle shows there's no hypertrophy and there is no dilatation. So it stays the same. But, and that was really surprising, when we measured the ejection fraction, and the fractional shortening is a related parameter, 2 of them, then we see in that mice a slightly increased contractility. With this small amount of mice, it does not get the significant threshold of 0.005, but that's a mathematical issue. Yeah. But we're getting very close already with this small number of mice. So that makes me very hopeful that we have something here at hand, which we can follow up now in mouse models of disease. And we have several mouse models for heart failure, and we will test it on that. So I would like to summarize this, my last slide before the typical ones, the summary slide. A class of small molecules, which we call SANVs, cause an allosteric inhibition of the enzymatic activity of NDPKs with some subtype specificity for B and or C. SANV3 is taken up in cells. Example, cardiomyocytes. It inhibits histidine kinase activity of the NDPKs. SANV3 in these cardiomyocytes acts context dependent. So depending on the subdomain, it's doing different things. In mouse cardiomyocyte, it increased the isoproternal induced cyclic AMP formation only in the Serca subdomain. In accordance, the SR calcium load and the systolic calcium amplitude is increased in the presence of SANV3. Also, in accordance with that, SANV3 enhances isoproternal induced contractility in explanted heart strips of rats, and might also increase the contractility in living mice. So I think that we have lead compounds here to develop new cardiovascular drugs. We have, of course, to increase the affinity, because 2 to 3 micromolar as an IC50 is still too low. If we want to really have a drug for humans, we need nanomolar binding constants. But I hope we can convince some chemists to do the work and help us to give the molecules a better binding affinity. So with that, I'm coming to my end, and I have to have a lot of thanks because it was a long journey. I pointed out the people who did the work, but it was a very close collaboration with the heart clinic in Heidelberg, especially Hans-Jörg Kippe, Johannes Barks, and also Matthias Deventer did a lot of work in that. These are people from my group who did different parts of that. I have to mention out especially Santosh Lomada, who worked with the inhibitors, Nadine Wolf, who did all the zebrafish work, Isam Abu Taha, who did the work on the heart. Oh, that's over. Sorry. On the hard work, and I had a lot of collaboration outside. I also have to think the fish, the rats, and the mice, and of course the people who gave us some money. Thank you very much, and I'm happy to take questions.
Okay, so we are open for discussion here. There is one question that I'm trying to transfer to you regarding, I may not have followed the full story. Does SAM3 increase phosphorylation of the L-type calcium channel in mouse cardiomyocytes?
No, it does not.
Besides acting on SEPCA and PLN?
No, we saw an increase only. Yeah, we saw an increase in the phosphorylation by SANV3 only if the agonist SANV isoproterminal is present, not basal. And we saw it only in the Serca subdomain on phospholamban. All the other targets, I had no time to show it, in the cells are not increasingly phosphorylated. That's why it's acting specifically in that subdomain.
And going on this question, I would like to ask something that is not a nerve expert, but if you think you have a drug, what would be the use of this drug? Or what type of heart failure
well,
suggest?
Well, heart failure is classified in several subtypes. So the most common is ischemic heart failure, but there are also dilated cardiomyopathy. But all in common they have at the end is that the beta adrenergic signaling cascade is put down. The beta receptors are overstimulated, so they are internalized, and the cyclic AMP production in the cell is permanently inhibited. And what we think is a chronic activation of GI takes a very important part in that. And NTPKC, that's the hypothesis. And we have some data pointing in that way, but they are preliminary, didn't show that. We can do the similar effects in cardiomyocytes on phospholamban phosphorylation with treating the cells with pertussis toxin. Pertussis toxin modifies GI and decouples it from other binding partners. And we get a similar action in this artificial setting. So what we think is NTPKC, especially in the heart failure situation, is constantly activating GI, and this is suppressing cyclic AMP. If we remove that part of that suppression, and not completely, will not go, but never drug should work complete inhibition, then we can raise that maybe to a level where the cyclic AMP is in a level where it can regain some contractility. All the efforts in heart failure therapy, which were treating cyclic AMP, increasing cyclic AMP levels, were targeting the phosphodiesterase so far. And this is very dangerous because then cyclic AMP is strongly increased, and then you got that calcium leak in the diastole and the tendency to arrhythmias. And that's why these substances are in the clinics only used for emergency therapy, not for other end therapy if nothing other is helping, but with a high risk. So I think we might have a modulator here, and modulators are always, let's say, more promising than these many targets we have. We could completely fail, of course. Yeah, it could end in nothing. I'm pretty, maybe within a year, I have to say it's not working. Yeah, but from that, what we have, I still think it's promising.
Just a curiosity. Thank you very much for this very nice talk. You mentioned the arrhythmia in excess of ventricular strength. Did you get in model animals an excess of arrhythmia Or which kind of arrhythmia?
Well, we have not tested that under the right conditions, I have to say. We have to test that in the failing heart animal models because the failing hearts are prone to arrhythmia. We didn't see in the healthy mouse, we have no sign of arrhythmia by our application of the drug. No sign at all. But we have
no changing concentration of the calcium.
this we can only measure outside of the animal because calcium measurements within is not possible. But what we're going to do is to implant in the animals transmitters which pick up the heart rate and also the beating over a long time, and then we would see extra beats, arrhythmias occurring or not occurring. That's how you do it usually. So, and I would not expect that something big is happening in the healthy animal, but the diseased animal, and there are several models you can use. You can use, for example, aortic bending to induce heart failure. You can make a model which is driven by chronic infusion of isoprenaline or isoprenaline and phenylephrine to mimic the high noradrenaline and adrenaline amounts seen in heart failure patients as a driver. Or you could use a myocardial infarction model in the mouse, which also will induce heart failure. But this is future work, and it's a lot of mouse work. We have done the animal applications. We got the animal applications granted. First, what we do is do a really dose finding study because we used only one dose. It was a shot in the blue.
At the moment, you may exclude that arrhythmia can be a secondary effect which will hamper the effect.
It will not exclude the arrhythmias at all as long as we have no data on that. So this is one of our most highly concerns that we could see something like that. But that we don't see that much change in the diastolic calcium under these artificial conditions makes me at least thinking that there's a way of getting there without the arrhythmias.
So if you would like to do, I go back to the same question, a trial in human, phase 1, what would you suggest? If you want to go to human.
Okay. What I would suggest what to do. So therefore I have to, before I go to phase 1, I have to have a complete toxicology in the mouse and maybe in another animal. I must have proof that it's working in the animal models, and I must have proof, some ideas about the pharmacokinetics of the final drug, not of this one. This one I will never put in a human. It's too low affinity. So I need a medicinal chemist or several of them, but because we have these different molecules which do the same depending on different subtype, and we have 3 more which are acting in the same way, I can give the medicinal chemist an idea how the structure must look like. And then they might, and what we are also doing with a group in Austria, we're trying to co-crystallize the inhibitor together with the NDPKC. And then we have a clear idea where the interaction starts. And based on this information, it should be possible to create molecules, variations with a higher binding affinity and also keeping the inhibitory constant. And then with such a compound, then you have to go through all these long-run toxicology, pharmacokinetics, and so on, until, and if this is all survived, then you can go in humans.
That was more or less okay. Grazie per questo talk, grazie Top. Ci vediamo al prossimo seminario. A meno che non ci siano delle domande dalla sala. Sono domande? Vediamo qui. Grazie ancora.